Hello, I'd like to welcome you to the Essentials of Intravenous Fluids. I am Allan Schwartz, DDS, CRNA. Before all of my courses, I present this disclaimer. The reason is that there's other learning materials besides the materials and the ways that I tell you, and I urge you to consult those other materials so that you get a fuller view of the topics that we're discussing. Let's talk about learning objectives. We're going to be talking about the cell, blood and plasma, common types of fluids for dental anesthesia, fluid administration, and armamentaria. Let's talk about the importance of fluids. Why do we use fluids in anesthesia? Fluids carry sedation medications to the patient's brain. That is where we are going to have the effects of our anesthetic medications. There are other receptors in the brain that produce sedation with midazolam, fentanyl, morphine, and ketorolac. Those are found otherwise throughout the system with narcotics receptors and affecting enzyme chemical reactions. One of the other things that we can do is to replace deficits due to NPO status. A patient usually goes 6 hours or longer with their fasting, and it affects the dryness or the wetness or the fluids that's within their bloodstream. And we can do calculations in anesthesia, especially for abdominal cases where a lot of evaporative loss is caused, to be able to replace those deficits and to maintain the patient's blood pressure. We're going to carry cardiovascular medications through fluids to their receptors. Examples are ephedrine, atropine, glycopyrrolate, epinephrine, and phenylephrine. Many of those are found in the drug toolbox. We're going to carry all medications to receptors other than those that we've talked about so far. Things like famotidine and diphenhydramine find their receptors and have certain physiological effects on the patient. Let's begin by talking about the cell. This picture is a really good one to memorize. This is an important picture, and we have our students learn this. We see that there's an arteriole that branches into capillaries, and then a venule where that blood is collected from having contacted every cell in the body. The arteriole, I teach in other courses, is where blood pressure throughout the whole system is controlled. By either tightening or loosening the arteriole, we can raise blood pressure or lower blood pressure. Notice that there is an exchange as we go further to the right in this diagram between the interstitial spaces, the spaces between cells, the cytoplasm, and even the nucleoplasm. There's constantly a cycle going back and forth. This fluid brings nourishment to the cell and takes away waste products. So by that exchange, nourishment can contain such things as salts, carbohydrates, lipids, proteins, nucleic acids. Notice that those are the building blocks of many of the molecules that the cells need or use to produce hormones and other secretions that they are programmed to do. This is a picture of the cell organelles. We won't ask you any questions on that, but you remember from your biology class that we, besides having genes within the nucleus, we've got such things as endoplasmic reticulum, where we package and secrete molecules, and we've got mitochondria that produce ATP to be able to provide fuel for the chemical reactions in the cell. If we were to enlarge the cell membrane, you'd notice that it would be what's called a lipid bilayer. We see what look like little balloons floating on the surface. That is the protein portion of the molecule. The lipid portion are what looks like the strings of the balloon, if you will, and there's two layers that are dissolved in each other. Notice that there's also proteins within this lipid bilayer. There's receptors. These proteins can function to provide gates to allow molecules in and out or ions in and out. So these proteins are integrated within the two-layered cell. The proteins, again, serve as passageways, and there's a lot of things that pass into and out of the cell that provide what the body needs and is programmed then to use. These proteins serve as receptors also for the various cells, and we have all talked about in our biology classes such things as receptors for the nerves. There's parasympathetic and sympathetic receptors, and we've got anesthesia medications that are synthetic molecules that stimulate those receptors, as well as many other receptors in the body. I want you to notice the cell environment. Notice that there's extracellular fluid and intracellular fluid, and that if we looked at the ion content on the outside and on the inside of the cell, they are completely different. Sodium is 142 milliequivalents on average outside the cell and only 10 milliequivalents inside the cell. You can look at all the other molecules there to see how different they are. Now, the most interesting thing of this is that Mother Nature wants that to be a balance. So we have to use energy to create this imbalance. It is not true of nature to have this kind of imbalance. If you recall the picture that I talked about earlier with the arterial and the venule, you notice that those arrows were moving in and out of the cells, the nucleoplasm, the interstitial spaces, and this Brownian movement is occurring even within 1/1000th of a second. The molecules move like crazy in many different directions. They're random directions, and Mother Nature wants a balance, so those molecules move in and out unless we apply a force to them. The force that we use, made by the mitochondria, is ATP, adenosine triphosphate. So there are 3 phosphate groups within ATP. And when just one of those phosphate bonds is broken by an enzyme, a tremendous amount of energy or force is released to be able to push molecules and cells toward something that Mother Nature may not want them to be. An example of this use of ATP is the diagram of active transport. So within the cell, there are 3 sodium ions that are pumped out of the cell, and as the cell pump continues to work, potassium is pumped into the cell, and that pump uses ATP, again, that releases a lot of energy. Let's talk about solids and liquids in human beings. If you were to look at an adult, especially these famous gentlemen, you'll notice that as an adult, they are 40% solids and 60% fluids or liquids. Now, 15% of that fluid is extracellular, and 45% are within the cells. So that's going to be the cytoplasm and the nucleoplasm. Children are different. Children are only 23% solid as opposed to the 40% in adults. And they're 77% liquids. 42% of it is extracellular, so there's a lot of extracellular water compared to an adult, and only 35% fluid is within the cell. You're probably wondering, what is the big difference between that? Well, the difference is a water-soluble medication like Versed will dissolve in that water, and it will be quickly removed from the circulation into the bloodstream and away from the receptors. So children will, if you will, redistribute that drug very, very fast. And there's other water-soluble medications. Let's talk about blood and plasma. If you were to take a test tube full of blood and put it in a centrifuge, you'd notice that as it would separate into layers, the plasma makes about 55% of the fluid that was totally there, and there's only 45% of red blood cells. So those are all packed in there. In the middle, floating between the two, is what's called the buffy coat layer. This is where white blood cells and platelets, which don't make up a whole lot of the constituents of blood, occur in that buffy coat layer. Let's talk about the purposes of blood. This liquid is going to transport nutrients and water via the plasma to the cells. We're going to transport oxygen to the cells. We're going to transport away waste products from the cells, and carbon dioxide, amongst other molecules, is brought into the bloodstream and taken away from the cells since they are waste products. We're going to also transport hormones, and this occurs very quickly in a lot of instances, to the respective receptors. One of my favorite monitors is the pulse oximeter, and there's no secret to it. It's measuring the redness of the blood, and the blood has to be moving or pulsing by the red and the infrared detectors. If you look at the probe that's there, it's kind of a buffy colored. You notice that there's the red signal that we can see, and there's an infrared signal, is the little bit lighter block next to it. And there's receivers or receptors that then receive the amount of redness, comparing the two frequencies of light that transmit an electric signal to the electric processor. There is a calculation that is made called the Beer-Lambert law, and then the monitor spits out the pulse signal and what percent of hemoglobin is saturated with oxygen. Red blood cells' only function is to carry oxygen. If we look at a single hemoglobin molecule, you can see that it can carry up to 4 iron molecules. The iron is in the Fe+2 state, so it's the ferrous iron. And as we know that if iron is oxidized, it turns red. So if we were to look at the redness of blood with only 1 oxygen molecule, it's not going to be as rusted or as red as if we've got the 4 fully saturated hemoglobin molecules. Let's talk about osmosis. We're going to talk about the words of osmosis. When you think of osmosis, I want you to think of the word tonic. And tonic is mostly salt, and there are some low molecular weight sugars that also produce tonicity, or the tonic of the fluid. So tonic and tonicity are 2 other words that we're going to use. If we define tonicity, it's the amount of salts and, again, the low molecular weight sugars in an intravenous fluid. And we compare that to the salts and the sugars found in the blood. So let's talk about some more vocabulary words related to osmosis. Let's put a cell membrane in the middle of the diagram. We're still in the bloodstream. There's hypotonic, hypertonic, and isotonic. Just to show you, we have osmoreceptors in our brain. It's right near the pituitary, and it's got a really good blood supply to be able to bathe the osmoreceptors with blood so that we can tell whether we need to drink water and be really thirsty, or whether we're adequate, or if we're overhydrated. This is the diagram that we talked about before. Notice that fluids are carried in the bloodstream and throughout the body. And then there's an exchange between every living cell all throughout the body. So a patient that is grossly obese, that fluid has to be brought and pumped to every one of their cells, or those cells will die. Notice that, again, those fluids bathe all of the cells. There's a constant contact and exchange with those fluids. Again, there's that interaction with the fluids and all the spaces between the cells. Let's look at a glass of water and talk about what isotonic means. Now I'm going to take the glass and I'm going to make it kind of into a bathtub, if you will, but it's still a big glass. And then I'm going to put the cell membrane between the right side of the glass. It's going to fill up the whole middle of the glass. There's no leakage. And the left side of the glass. Just to show you the extracellular fluid and the intracellular fluids and how they're separated by the cell membrane. So let's start an IV in the patient and run some fluid through it. This is from that first slide we looked at of the cell membrane, showing you the basic concentrations of the ions in sodium, potassium, chloride, glucose. And then again, there's the intracellular fluids on the inside of the cell. So we've hooked up the IV, and we're going to run an isotonic solution. So iso means equal. Let's see what happens. We're going to come down to the capillaries. There are two isotonic fluids that are commonly used in sedation or dental anesthesia, and those are LR, lactated ringers, and normal saline. So those are going into the bloodstream. These two sides of the system are in equilibrium. If you look at the salt shakers, they're equal amounts of salt in both the extracellular side and the intracellular side. So the fluids are exchanging across the membrane, and they're very happy. They're flowing very freely because they are of equal salt or equal tonicity. If we have a lot of salt that we've ingested, it of course makes us very, very thirsty, and that's because of our osmoreceptors. Notice that the salt shaker on the IV or the extracellular side is really salty compared to the normal salt shaker on the intracellular fluid side. We have a fluid, it's called hypertonic saline. Notice that the letters on the IV bag are in red, and it is 23.4% sodium as opposed to 0.9% sodium in normal saline. We call normal saline because it's the normal salt content within the cell. Notice that hypertonic saline is available in 2%, 3%, 5%, 7%, and 23% sodium within the hypertonic saline compared to normal salt or normal saline, which is 0.9% sodium and chloride. Notice that these have to be given, as I said, with a pump, and the patient is gravely ill for us to need this to be able to pull water out of their brain or out of their system and to hopefully get them back into a more stable state. Let's talk about hypotonic. Notice on the extracellular side that we've got a small salt shaker, and then on the intracellular side, we've got a large salt shaker. So the salt on the extracellular side is too small or too reduced in concentration compared to the salt just even within a normal cell. And there are hypotonic intravenous fluids that we can give the patients. Examples are half normal saline, where about half the concentration of sodium as normal saline, and then there's dextrose 5% in water, also called D5W. So these are two hypotonic solutions. So what's going to happen is, since there's more salt on the intracellular side of the cell, fluid is going to try to go and does go across the membrane, the cell membrane, the lipid bilayer, and will try to dilute and make equal the salt content within the boundaries of the cell membrane. Here's an example if we give an isotonic solution into the extracellular fluid. Notice that the shape of the erythrocyte, it's called a biconcave disc, and you've seen those big discs that we've got in the background of this photo. And salt are exchanging very well between that cell membrane of the red blood cell, and the cell is its normal size and shape, and that's the way it works in the body. It works best to be able to exchange oxygen and carbon dioxide when it's this biconcave disc shape. When we give a hypotonic solution, notice again the water is going across the membrane, it's not exchanging equally, and we see that the cell then becomes enlarged, or if you will, it becomes blown up. Notice that the salt content and that extracellular fluid in that middle picture, there's not a lot of salt there compared to the intracellular side. Slide 78 shows you the complete opposite. We've got high salt, so there's a lot of sodium molecules in the background, and the cell water, the intracellular water, will come out of the cell membrane to try and equalize that. That's a force of nature, and it will shrink the cell. So neither the hypotonic red blood cell nor the hypertonic red blood cell can function in the body. Another set of pictures for the hypertonic, isotonic, and hypotonic extracellular fluids being given to the patient. With a hypotonic fluid, we can blow up the cell so severely that it will break and burst, and we'll get the hemoglobin contents released into the bloodstream and then carried away and metabolized and excreted in the body. It's either in the stool or we may urinate it out. Again, shrunken cells in that hypertonic environment don't work. It's an odd-shaped cell. It can't function getting into the capillaries, into the picture that you memorized of the arterial, the capillaries, and the venule. These are a list of the 3 available isotonic fluids. Notice again that we've got normal saline, lactated ringers, and I'd just like it to show you that we've got a solution that we use in cardiovascular surgery called Normosol. It is also isotonic. If we want to add dextrose molecules because a patient's got low blood sugar, say they took their insulin and we've got to get their blood sugar boosted up and they're going in for surgery, we have a D5-LR and a D5 normal saline solution available that will provide that dextrose in an isotonic fluid. In our emergency drug kits, we also have dextrose 50 grams within a bottle that should be in your emergency drawer. This shows you the recipe for normal saline. It just shows you the number of milliequivalents of the different ions and what the pH of the solution is. Here's the recipe that Dr. Ringer came out with when he introduced and developed lactated ringers. Lactate acts as a buffer to buffer between acid and base in the bloodstream. Notice also that from this recipe that there is calcium in LR. This calcium will inhibit the molecules that cause the blood to remain unclotted if we were going to give blood products to a patient. So the calcium will undo that unclotting mechanism. So we don't use lactated ringers when we're giving blood or blood products to a patient because of that calcium. Here's hypertonic saline. It's got a high sodium content compared to normal saline. I just want you to be aware that hypertonic saline is extremely rare. We have to go to the pharmacy with an order to be able to pull this out of the pharmacy. The patient is usually gravely sick with some fluid accumulation that needs to be brought out. So we're going to use osmosis to be able to remove the excess fluids in a patient's system. Usually these are ICU patients and they're very sick. So it has to be given through a pump and it has to be really carefully monitored. Here again are those commonly available hypotonic fluids. We've got D5 in water, dextrose 5% in water, half normal saline, and there are irrigation products we use called sterile water. It looks just like an IV bag, but we would never ever want to give that to a patient. I want you to be aware that the 2 on the left, the D5W and the half normal saline, are given to patients, but again, it's pretty rare. we would use probably normal saline because of its lower cost to give our patients that are having sedation anesthesia. It's going to be important for you to know how we gain and lose water, how it happens normally, especially when the patient's been NPO, again, nothing by mouth. The ways that we gain water are by drinking water, ingesting foods. The oxidation of food will produce water as a metabolite. The oxidation and the aging of body tissues. We're going to lose fluid by perspiration, breathing, respiration. If you've been in a cold climate and you've not gotten the heater working in your car yet, you can see the humidity that accumulates on the windows because the windows are so cold and they condense the fluid out of our breath. By urinating, we lose fluid. And while I'm thinking of it, before you take the patient back to the operatory, I always make sure that they pee before we start their sedation, start to get their IV in. We've got everything ready and we're going to be doing moderately long dental surgery. We're going to lose fluid that is contained within stool from bleeding and vomiting. And just being NPO. And an open wound, since it's open and doesn't have any tissue over it, will also lose water. I would know each of those and make sure that you've got those learned. The ways that sedation dentistry causes fluid losses are due to the surgery. Again, we're going to have exposed tissues and bleeding loss. From illness, should the patient get sick and vomit during the surgery, if we have to get them an emesis basin or we need to get them over to the bathroom real quickly. And just anesthesia itself, that NPO status really affects the amount of fluid in a patient's bloodstream, and they can have negative consequences because of that NPO status due to the dryness or the fact that they don't have enough fluid in their bloodstream. It can precipitate dizziness and nausea. So anesthesia patients are going to be what we call behind in their fluids right at the start of the appointment due to the pre-op fasting, being NPO. And I like you to look at the guideline that we all follow in anesthesia regarding NPO status. Notice that clear liquids are a 2-hour period of time before they've had the clear liquid and when we can start their sedation. If they've had any kind of a light meal, we go 6 hours. A light meal consists of, say, dry toast, maybe a hard-boiled egg, or something on this order, something without any fat in it. And a heavy meal is a new guideline that came out in 2017, and it is defined as just a regular fatty, heavy meal that a person would eat. Anybody that's had any edibles with the advent of the use of marijuana, we go 6 hours for their NPO status for that. If they vaped, we usually go about 2 hours or so, but it's kind of a judgment call at that point. Again, the patient is dry. We've got to try and replace some of that fluid. I unfortunately have had the experience when I've tried to replace a lot of the fluid in the dental chair that the patient has peed in the chair. So we've got to be really cautious when we're giving fluids to a patient. We can't recover them the way we can in the OR and try to get them out of their dry NPO status. So in the OR, we try to fill up the tank, and that's what I'm asking you to do with the judicious use of fluids. If need be, the patient can be awoken or made sure that they're stable and balanced and get them up to go to the bathroom, say after about either an hour, hour and a half, or by the latest 2 hours, since they don't have a Foley catheter in and we're not giving them tons of fluid, but there's times that you're going to probably want to just make sure that they're comfortable and that they don't pee in the chair. So again, filling up the tank means opening up the IV fluids. and giving the patient some fluid. If a patient was hypotensive and we needed to get their blood pressure up quickly, be it in the dental operatory, I would not hesitate to open the fluids full blast and have another bag ready to go. And again, assist every patient into the bathroom. They might be dizzy due to their fluid, due to the sedation medications, and we want to make sure they don't hurt themselves. So if too little fluid is given, we will notice, we'll see this with the NPO status, that heart rate will be increased for sure. So they have tachycardia. Their blood pressure can drop. The heart rate increases to try and get blood to the brain. And if their blood pressure drops, the heart is going to go even faster. So we are going to want to open up the fluids in these patients. We have got to keep their blood pressure at a homeostatic level. In other words, a level where they normally live is considered to be a normal blood pressure for them. If too little fluid is given, again, the patient could lose equilibrium or faint and vomit. Here are the emergencies, one of our other courses that require fluid therapy. Notice that there is hypotension, cardiopulmonary arrest, allergic reaction to drugs, angina pectoris, possible MI, possible myocardial infarction, emesis with potential for aspiration of vomit, convulsions and seizures, and then oversedation. As we're making up the administration set and getting ready to attach the bag, make sure that the bag is not punctured. The bag on the right, the LR bag, is wrapped in plastic, and there is a sterilization process that the manufacturer does, and it creates condensation of water in these plastic bags. So don't worry if you see that there's water that comes out as you tear the bag, the plastic outer bag open. We've got to also be aware of contaminated IV bags. They should not be frosty. You should not see any particles in a IV bag that we use for sedation. Again, any particles in the fluid, if it's a frosty colored, and if the bag is expired, we want to not use that. And you have to make sure that you're using the correct fluid for the patient's needs. If someone has some kidney disorders, we would probably go to sodium chloride, and we would reduce the amount of fluid that we would bombard that patient or give IV. So normal saline we would put on a really slow drip if someone's got kidney problems, chronic kidney disease, something on this order. Here are our references for the course. Thank you very much.